A rat sits alone in a cage, two bottles in front of it, one filled with plain water, one laced with a highly addictive drug. The rat sniffs both, then it chooses the drug, over and over again, until it dies. For decades, across countless labs, the result was always the same. Scientists held up the results and said, "This is what drugs do. Once exposed, you lose your free will." For most of the 20th century, this narrative formed the foundation of our understanding of addiction. It shaped slogans like, "Just say no," framed addiction as a moral failure of willpower, and drove decades of harsh policy. But it was only half the truth. The man who discovered this was Bruce Alexander, a psychologist at Simon Fraser University in British Columbia. After years of studying these experiments, he noticed a glaring flaw. The rats weren't just testing drugs, they were in solitary confinement, trapped in small metal boxes with no stimulation, no friends, and nothing to do but take drugs. This realization prompted Alexander to ask a paradigm-shifting question. What if the rats weren't choosing drugs, but simply choosing escape? To test this theory, Alexander and his colleagues began building something unprecedented, the rat park. This 9-square-meter enclosure was nearly 200 times larger than a standard lab cage, effectively moving the rats from a prison cell into a sprawling neighborhood. The walls featured painted landscapes, and the floor was filled with cedar shavings, climbing platforms, and tunnels. Most importantly, it was populated with a diverse mix of male and female rats, a real, functioning society. Observing from above, researchers witnessed everything absent from traditional trials. The rats played, fought, groomed, and slept in piles. They were finally living. Then, Alexander introduced the two bottles, plain water and the drug, that had consistently induced addiction in isolated cages. Traditional science dictated they would all become hooked, but when the data came in, it contradicted 40 years of research. The socially enriched rats mostly ignored the drug water, vastly preferring the plain water. Week after week, the pattern held. This alone contradicted 40 years of research. Thinking the drug might just taste bitter, Alexander sweetened it, combining two highly addictive rewards into one irresistible bottle. Pleasure layered on top of pleasure. If anything was going to break them, this was it. Yet, even when offered this irresistible blend, the rat park colony still favored plain water. While they occasionally sampled the sweetened drug, their consumption remained a mere fraction of the levels seen in isolated cages. The difference remained staggering. Alexander increased his scrutiny. Maybe something subtle was being missed. Maybe the rats were using the drug in hidden ways. Maybe addiction simply required more time. So the experiment continued. Weeks became months. The pattern refused to change. Again and again, the same conclusion emerged. The rats with rich social lives consumed far less drug water than the isolated rats. The implication was becoming increasingly difficult to ignore. The drug, its chemistry, and the dosage remained exactly the same. Only the environment was different. And yet the behavior had changed dramatically. For decades, scientists had looked at addicted rats and concluded, drug causes addiction. But Rat Park was beginning to suggest a different possibility. Isolation breeds desperation, driving creatures toward any available relief. That idea was dangerous. It threatened something far beyond a mere laboratory theory. It meant the flaw of addiction lay not just in the chemistry of the brain, but in the design of society. But Alexander still wasn't satisfied. Critics could still argue a crucial point. Perhaps the socially enriched rats never became addicted in the first place. Maybe they had simply avoided becoming addicted in the first place. To answer that question, Alexander designed a final, definitive experiment, one that would become the study's most famous and perplexing phase. This time, he wouldn't start with healthy rats. He would start with addicts. He took a new group of rats and placed them in complete isolation, mimicking the standard addiction experiments of the past. For the next 57 days, they had no choice but to drink the drug water. By the end of this forced chemical dependency, the rats were hooked by every established measure of addiction science. Their very nervous systems had reorganized around the substance. By now, they were exactly the type of addicted rats standard science relied on. Then came the real test. The rats were transferred into Rat Park. Crucially, the drug water remained freely available. The researchers weren't forcing withdrawal or offering a cure. They were simply giving them a choice. Alexander later called those first days in Rat Park the most interesting phase of the project. The newly arrived rats behaved differently from the regular residents. Instead of isolating themselves, they eagerly explored the vast enclosure. Despite brief chases and minor scuffles, they quickly began integrating into the complex social structure. Meanwhile, researchers closely monitored the drug water. Under the traditional chemical theory of addiction, These severely dependent rats should have kept drinking heavily. There was simply no reason to expect a sudden detox. But that wasn't what happened. While the animals still sampled the drug water, their consumption plummeted as they increasingly preferred plain water. The transition wasn't immediate, but unfolded gradually over the following weeks. They endured the painful withdrawal of their chemical dependency, actively choosing discomfort over the drug. Instead of hovering around the drug bottle, they engaged with their surroundings, exploring, sleeping in groups, and participating in normal social behaviors denied to isolated rats. Because the drug remained freely available, the implication was inescapable. Chemically dependent animals, placed in a meaningful environment, voluntarily detoxed. Alexander had proven a profound truth. The isolated rat's craving was never really about the drug. It was about the cage. The drug was simply the only thing in that cage that felt like anything at all. A few years after Rat Park, another radical observation emerged. But this time, they weren't looking at rats in a lab. They were looking at American soldiers in Vietnam. By the early 1970s, heroin use among U.S. troops had reached epidemic proportions. Military surveys revealed a staggering reality. A massive number of troops had become fully addicted to heroin while deployed. Terrified that these veterans would bring their habits home, U.S. officials braced for an unprecedented wave of domestic drug addiction. But as the war drew to a close and the soldiers returned, the anticipated crisis simply never materialized. One of the most influential follow-up studies was conducted by psychiatrist Lee Robbins. Her team tracked returning veterans who had used heroin in Vietnam. Experts assumed the addiction would follow them home. Instead, the vast majority simply stopped, and they did it without years of rehab or forced withdrawals. The drug hadn't changed, nor had human biology. The only variable was the environment. Though Robbins was studying veterans and Bruce Alexander was studying rats, their work ultimately pointed to the exact same question: what happens when a person's environment changes? In Vietnam, soldiers faced intense stress, constant danger, and deep isolation. Then, almost overnight, they returned to the safety of their homes, jobs, and communities. Once the nightmare of the war was replaced by a normal life, the addiction simply vanished. Alexander published his findings in the early 1980s. The scientific establishment largely ignored him. The results did not fit the model. They contradicted too much funding, too many careers, too many years of institutional momentum. His grant applications were denied. The mainstream addiction literature kept citing the cage experiments as if Rat Park had never happened. Yet the findings refused to disappear, because beneath the rat experiment was a much larger question, one that wasn't really about rats at all. For decades, we built our entire understanding of addiction around the most artificial possible conditions. We took a social animal. We stripped away everything that made its life worth living. We gave it one source of relief. And when it took that relief, we pointed and said, "Look how powerful the drug is. This is addiction. This is the enemy." If the problem is the drug, the solution is to remove it, punish the person holding it, build higher walls around it, declare war on the substance. But if the problem is the cage, then removing the drug changes nothing. The person is still in the cage. The emptiness is still there. They will find another way to fill it. Alexander was not the only one who reached that conclusion. A journalist named Johann Hari spent years revisiting the research and the broader science of addiction. His conclusion echoed what the rats had already shown. The opposite of addiction is not sobriety. It is connection. Not the absence of a chemical, the presence of a life. None of this erases the biology. The chemistry of physical dependence is real. For some people, with some substances, the physiological grip is fierce in ways no amount of social enrichment can simply dissolve. But the rat in a cage experiment never captured that complexity. It captured something far simpler. And yet, we built a century of policy on that foundation. And the most honest question we could ask about addiction, about all the ways people vanish into things that hollow them out, is not what is wrong with them. It is understanding the cage they live in.